My name is Dale Yuzuki. I want to welcome you to this section on new approaches in the genetics of disease. And today I'm here to talk about advances in high throughput proteomics using NGS as a readout. I am a member of the marketing team at Olink Proteomics. And you might say, well, this is a genetics of disease. Why are we talking about proteins? Well, I'm glad you're here. Today's outline will talk a little bit about Olink in the context of the challenges of 21st century healthcare, and there are plenty of challenges that remain. In particular, one of scale, because we're used to looking things almost like genomics in 2000 or 1999, when you were looking at a single a nucleotide polymorphism on a scale of dozens or maybe 100. And you'd say, well, how can we take a look at 100,000? How can we take a look at a whole exome's worth of mutations? We have a similar challenge of scale with protein analysis as well. And then I'll finish with 3 practical applications of this technology, which is looking at Alzheimer's, looking at multiple sclerosis, and looking at drug discovery. Now, a little bit about Olink. So Olink is a worldwide organization with many top customers, founded in Uppsala, Sweden, and then having presence in Boston and the Far East, in particular Shanghai, Tokyo, and Singapore. Another thing I'd like to point out from this slide is there are over 800 peer-reviewed publications on the platform. Olink got its start in 2004. However, it was not until about 2015, 2016 that Olink started commercializing a 96-plex protein assay that we currently call Olink Target 96. This climb in peer-reviewed publications has recently spiked upward with greater and greater interest in circulating proteins in serum and plasma as biomarkers, and in particular, most recently, the combination of genetic variation data with circulating protein biomarker data for drug discovery and for looking at mechanisms of disease. The last thing I'll point out on the far right is consortia, in that there are several global consortia, 1 or 2 of which I'll mention in this talk later. And as far as this particular slide, global relationships, local laboratories, and proudly Swedish. The term proudly Swedish may be a bit of a misnomer, because I think Swedish folks, in my understanding of them and my friendships with them, they tend to be a self-effacing group. But nonetheless, Uppsala may sound familiar because, as a matter of fact, it was in Sweden, in Stockholm, that ELISAs were first invented back in the 60s. It is where cross-linked Cephidex was invented, forming a company called Pharmacia. For you old-timers out there, you may remember Pharmacia in the late 90s for bioprocessing and chromatography. Now, Uppsala still has a large presence from Cytiva, which is the GE Healthcare, right, which acquired Pharmacia, Amersham, those kind of companies. And then they still have a large presence in Uppsala. I was just there last week, and Uppsala is just a small city, relatively small, 150,000 or 170,000 or so people, but it's remarkable in terms of the amount of protein expertise, the amount of understanding of different technologies. And as you can see from the icons and below, we have customers global, worldwide. Now, this particular slide illustrates the picture of 21st century healthcare. And you don't have to go back, you have to go back 20 years to essays that were written in 2000 and 2001 about the promise of the Human Genome Project, and how it was felt that the diagnosis and the development of medications was going to just revolutionize healthcare. We're going to cure diseases. We're going to be able to understand so much through the Human Genome Project. Yet, 20 years later, or 22 years later, I mean, the map was announced in the Lincoln Room of the White House. You can look up that particular speech. It's remarkable. You have Craig Venter sitting on one side of Bill Clinton. Then you have Francis Collins sitting on the other side of Bill Clinton. And Clinton talking about, the most wondrous map that mankind has ever produced, the map of the human genome. This is the year 2000. And then naturally, 2001, the two landmark papers in Nature and Science, with the first draft of the human genome. It was very, very exciting, certainly. For those of us who were involved in science at that time, extremely exciting times. But what has happened in the last 20 years in terms of all the genomics that has taken place? There have been tens of thousands, right, of genome-wide association studies to look at inherited risk for common and for rare disease. And you can say, the Human Genome Project has certainly benefited monogenic Mendelian disorders. And so we're looking at, out of, I think, something like 7,000 in OMIM in terms of Mendelian disorders, inherited disorders in man. Only about half of them have a clear genetic, we've identified the genetic component, and then a fraction of that are druggable, and or there's some sort of therapy you can do around it. So certainly in the realm of rare monogenic disorders, the Human Genome Project has been just wonderful. Another area where the Human Genome Project has revolutionized healthcare, I'd point to cancer. And the development of targeted therapies, about 42% of phase 1, phase 2, and phase 3 cancer drugs that are going through clinical trial improvement now are targeted therapies. That is, looking at the molecular characterization, mainly DNA aberrations, from the patient's tumor samples to guide treatment and having therapies against those molecular alterations. So that is another area where the 21st century healthcare has been fundamentally changed through the advent of genomics. I'd say a third one that impacts sort of the modern person. I remember certainly when I had my first child, we had to go through amniocentesis. It was invasive. We had the known risk of about a 1% risk of miscarriage in doing this diagnostic test for the health of the fetus. Now it's been completely replaced through non-invasive prenatal testing, or NIPT. And this is where circulating blood DNA, right, is able to pick up on shedding fetal DNA and assayed for trisomy. Now you look at moderate in terms of RNA, and naturally, right, with the advent of RNA vaccines, that is a huge opportunity and breakthrough, right, through this pandemic. There are certain RNA-based drugs, either RNA interference or using other RNA mechanisms, short interfering RNAs and what have you. However, the learnings from all the RNA work and RNA-seq and transcriptome work has been moderate, right, in terms of either RNA-based diagnostics or RNA-based therapeutics. Now, looking at proteins, naturally, proteins are the workhorse of biology. When you think about everything from structural proteins to enzymes that replicate DNA, that of course transcribe it into RNA, and of course, as far as the proteins that are doing anything in a cell, right? This is where the action occurs. And when you look at, for example, FDA diagnostics, there are over 200 diagnostic tests that are based around proteins in terms of measuring protein levels. So, and then even the vast majority of drug targets, okay, in terms of pharmaceuticals, they are working against protein targets. That's on the order of 95, 96% of all drug targets are proteins. So as far as Olink, right, why scientists use Olink products to measure proteins, in the upper right and the darkest section is a better understanding of biology. Scientists looking at just circulating proteins can understand biology on a real-time basis. They are looking at, panels of proteins for people that are risk for heart failure. We have customers who are working in diabetes, and they are understanding the risk for kidney failure through their samples and through their sample cohorts. By circling around on the right, predicting disease and outcome, right? So our pharmaceutical customers are looking at being able to determine the outcome based upon blood biomarker signatures. Patient stratification, there's an example of interstitial lung disease where you can determine which patients will actually have progressive fibrosis in an interstitial lung disease example, and other patients that will not, and using that to stratify your patient population in the clinical trial has the potential to save about 1/5. You can use 1/5 of the patient samples, only 20%, saving 80% of the value of the cost of doing that particular trial. There are surrogate markers for safety and efficacy, naturally of great interest to drug discovery, and wellness from health to disease. There have been some fascinating wellness longitudinal studies. That is, taking a population of individuals, simply taking their blood at regular intervals, and then being able to track the individuals who remain healthy and the individuals who declined into different kinds of disease. And these kinds of longitudinal studies are extremely, extremely powerful. As one example, researchers at the Institute of Systems Biology in Washington State had a spin-out company called Araval. They followed several hundred individuals who were healthy over a course of several years, and they were regularly taking blood samples. Well, 3 of these individuals developed advanced metastatic cancers. And it's awful, right, that when you think about the individuals involved. However, the fact that these individuals who got these advanced cancers and were diagnosed with these advanced cancers, you had pre-diagnostic cancer samples. You knew this person was developing, say, stage 4 pancreatic cancer, which there are very, very few reliable biomarkers, and of course, very, very poor treatment and prognosis. The fact that they had the pre-diagnostic blood samples from people who ended up getting a horrible disease, they can now look into those pre-diagnostic samples and determine what was different. And the paper published in Nature Communications revealed 3 remarkable markers that were very solid and very statistically significant markers that had not been characterized before as early detection markers for advanced metastatic disease. And so not only was it pancreatic cancer, it was also lung cancer and one other. Now this slide describes the complete drug development process, which our customers in pharma take advantage of. They use it for discovering drug targets, and that first minor point in terms of protein quantitative trait loci, they're actually identifying proteins with strong evidence of causality using the genetic background of the individual, the circulating level of the protein, and the disease state in order to ferret out the causal protein for that disease state. It uses a method called Mendelian randomization, and it is an extremely powerful tool. On the discovery side, Olink now is able to detect 3,000 protein biomarkers, and it is quite a powerful tool to be able to have this broad screen of understanding the underlying biology. As a for example, right, where in preclinical research, and you really want to understand the mechanism of action in terms of the particular molecule in question for therapy, you're able to see the perturbation in the circulation of the circulating proteins and being able to match that against the model in existence of what the mechanism of drug action is. You can see through phase 1 with safety, phase 2 efficacy, the large majority is something on the order of half, right, of all clinical trials fail just due to efficacy, being unable to show that the drug in question that's in development is able to provide benefit to the patient. An interesting story on the post-market surveillance. Even after approval, we have customers who take a look at those samples that were collected during the clinical trial to give them clues as far as how that particular drug that's on market for one indication can be used for other indications. And you might say, well, how does that work? You say, well, okay, they see the pattern, right, of the proteins that are perturbed. They understand more of its mechanism of action. They say, well, these particular proteins are being downregulated and these other proteins are being upregulated. That seems like an ideal candidate for this other disease that we hadn't thought about before. Then they can take the existing approved drug for one indication and go back and say, let's take a look at this and start at phase 2 because we already know the safety profile of this particular drug. Now we're going to see if it's efficacious against this other indication. So this can be an extremely powerful tool in the hands of a pharmaceutical customer. Like I mentioned, in the discovery side, Olink now has the capability to look at 3,000 proteins. And we're also then to verify in larger numbers of samples using panels of 96 proteins. And in terms of down to the laboratory developed test and a future in vitro diagnostic, we have a custom panel that's only 21 proteins. We have a customer in San Francisco called Octave Bioscience, and they're aiming to revolutionize the way multiple sclerosis is handled and taken care of. And the beauty of what they're doing is that they have developed a screen. First, they screened 1,400 proteins against their patients with MS of different statuses. They're able to develop a multiple sclerosis disease activity test that's able to better characterize patients that have subclinical progression. That is, the current status of multiple sclerosis is you take an MRI with a tracing agent, and oftentimes it's catching the progress of multiple sclerosis after it's progressed too high. What they're doing is they're finding a lower level of disease progression using a circulating biomarker assay developed in conjunction with Olink in terms of discovery and then validating their results in both analytically for laboratory developed tests and with multiple sclerosis patients. The historical challenges in proteomics has been a lack of sensitivity and the lack of throughput. So this combination, you can either have high sensitivity and low throughput, or you can have high throughput and low sensitivity. On top of this, high sample consumption, for example, a competing platform may only offer 10 proteins at a time, but it consumes 50 to 100 microliters of sample. That can be an issue. Dynamic range, well, proteins have a 12 logarithm range of high abundance to low abundance proteins. The data analysis, the cost, and particularly the lack of specificity. It's not unusual for someone to do a multiplexed bead-based assay and have to do experiments in duplicate or triplicate just because of the variability in the results. I'll go into how the proximity extension assay works, but we've overcome these technical challenges with a technology that has very, very good specificity due to the use of 2 antibodies instead of 1. extremely high sensitivity down to the femtogram per mil range. And on top of that, there's this dynamic range that covers 10 logarithms from femtograms to milligrams. We use very small amounts of sample. It's really less than 10 microliters is needed for a 3,000-plex assay. Very high throughput. The assay is set up in 96-well plates. It is designed with high throughput and liquid handling automation in mind. It's the scalability, like I just mentioned. Validation, if you go to olink.com, you'll be able to find the validation data for each of our 3,000 targets. And there's over 800 peer-reviewed publications, like I mentioned. So I'd like to share with you a movie that illustrates the proximity extension assay via an animation. And so what we have is matched pairs of antibodies that each have a unique bar-coded DNA strand covalently attached to it. When you have two correct antibodies recognizing the target, naturally the Watson-Crick hybridization can take place. We use an enzyme to extend the DNA, which adds to specificity, and this DNA strand that's been extended now is the template for a readout. Here in this case, the readout is real-time PCR. Now you can understand the same process moving forward with NGS, where instead of the count on an Illumina sequencer. So this is just showing sequencing by synthesis. But nonetheless, we have a digital signal that corresponds to the protein concentration in each sample. And because of the way we're able to barcode the identity of the antibodies, right, in terms of the antibody pairs, we're able to identify the protein. And due to other barcoding that happens in the well, we're able to identify the sample in this particular library of molecules. And as far as then our ability to screen lots and lots of proteins, right? We have screened over 10,000 assays, all right, a generic pool of antigens. There's a second pool with expanded sensitive antigens. And then we verify, right, against cross-reactivity. So this basically a library of 3,000 different protein assays. We know exactly what we're measuring, which is an important point because there are alternatives on the market that just show patterns or will give you signal without the full understanding of what exactly it is you're measuring. However, here in the Olink case, we have 3,000 whose identity is very well characterized. Now, this next slide illustrates the plasma proteome, and I mentioned there were some 12 logs of dynamic range between the most abundant proteins on the upper left and then the least abundant proteins on the lower right. And as the illustration shows, our scope covers the entire range of traditional top-down proteomics using mass spec, as well as the low abundant proteins that traditionally have been the province of immunoassays. And the way we're able to do, well, I think one of the interesting things from this chart is the top 22 proteins in this chart comprise 99% of the mass of proteins in a particular patient sample. So it's a really difficult problem, right, to be able to either deplete those proteins or verification data of correlation to clinical immunoassays for 3 common markers across different platforms from Roche and from R&D systems, and they correlate very, very well in terms of, understand, the Olink assay was 1 of 96 in this particular example, and we're looking at 96-plex, and the measurement of these 3 different proteins in the context of singleplex ELISAs. And naturally, ELISAs are considered the gold standard. The correlation to mass spectroscopy, you see how, or spectrometry, you see how close it is in terms of overlay, and this represents 170 different samples. In terms of platform, like I mentioned, we have a 3000 on the high end for discovery. We got panels of 96 and one for cytokines at 48, and a real-time instrument that's associated with the midplex, or this 96 multiplex immunoassay, as well as a custom focus for 21, like I mentioned before. Just a little bit about the panels, right? We have these 3,000 divided into 8 panels of 384. And then we have 14 human panels plus a 15th for mouse of 92 assays apiece. This is what we call Target 96. I think one aspect of Olink that is not mentioned very often is the fact that we have a data science team that is dedicated to supporting our customers for the data analysis, from quality control through data visualization and statistical analysis. Also, for customers who want to do their own analysis, we have R packages as well as online tools for statistics and for generation of nice visual plots that you can just drop into a manuscript or publication. In this slide, you can see the 19 protein model for Alzheimer's disease classification outperforms the classic markers. And at the top, you can see the classic marker ATN in yellow for the discovery cohort. That particular AUC was only 87%, whereas the 19 proteins offers a 98% AUC, which is remarkable, right? And not a significant difference when you add the ATN plus the 19 proteins. On the bottom, validation cohort, similarly, 88% AUC for the ATN, but you have a 96% AUC using the 19 protein model. Remarkable. This is, I believe it can be a game changer for Alzheimer's disease detection, as in particular early detection and stratification of the patient population. This is the most comprehensive characterization, looking at 1,400 plasma proteins for Alzheimer's disease, and being able to discriminate patients from healthy controls, right, in 2 separate cohorts. And it also offers opportunities for drug development. I mentioned in my prior remarks about Octave. We'll just cover this very, very briefly. As far as what Octave did was look at 1,400 proteins, drill down into an 18-plex disease activity test that covers 4 key aspects of the disease biology. And their particular results, like I mentioned, they'll be launching this particular test to the multiple sclerosis community later this year, and it'll be very, very exciting to watch. The last example covers a consortium called SCALLOP. This is Anders Malerstig from Pfizer and from Karolinska Institute, who is organizing an effort called the Systematic and Combined Analysis of Olink Proteins. And this is combining genomic variation with disease status and the circulating proteome. And if you look on the cartoon on the right, you have the disease individuals, you've got the proteins, the disease, and the genetics, and then you have drug targets. So the goal of SCALLOP is to combine genomic variation information of the individuals with their circulating proteome, and then to be able to determine strong candidates for drug targets. And this particular paper, published in late 2020, genomic and drug target evaluation of 90 cardiovascular proteins in 30,000 individuals. What this study did was take a look at a single Target 96 panel and combine 15 different collaborators within the SCALLOP consortium. And what they ended up finding was truly remarkable. This is a busy slide, but nonetheless, The opportunity was to identify protein quantitative trait loci and use a method called Mendelian randomization to provide strong or intermediate causal evidence for new drug targets. And as you may or may not know, it takes a long time to develop a drug today, and it's extremely expensive, about $2.6 billion, all right? What is the results? The results is in green. They identified 11 proteins not previously targeted with causal evidence of involvement in human disease, right? This is causative proteins that are new drug targets. They identified in blue a whole collection, okay, of proteins for target validation. And in purple, they identified targets for repurposing and target-mediated safety. So for drug discovery, this is, I mean, one paper cannot capture all the value in it, all right? They looked at 30,000 individuals, 85 proteins, and 450 pQTLs. And each of those 85 proteins, right, had positive associations for causality of different disease types. So I invite you to go ahead and take a look at this paper, take a look at the supplemental information, take a look at Table 7 in the supplemental information that gives you all the details of these 85 different genes. And lastly, we'll mention the Pharma Proteomics project. This is a project in collaboration with the UK Biobank where there were 10 pharma partners who have sponsored this to take a look at 50,000 samples of the UK Biobank. Again, these are individuals from 2006 to 2010, a total of 500,000 individuals who have consented to not only have their genome sequenced, but also their blood samples and their health information recorded by the UK Biobank. So as time goes on, the UK Biobank becomes more and more valuable as these individuals, unfortunately, will get sick and unfortunately, right, will be treated. But nonetheless, the genetic backgrounds will be known, the blood samples will be taken. Like I mentioned, those, pre-diagnostic cancerous samples will be obtained, and it's going to be a remarkable, remarkable project. So the UK Biobank has already moved on to the Explore 3K. They're looking at 53,000 samples from the UK Biobank, okay, with 3,000 circulating proteins, where the genomic information is known, where the circulating proteome information is known, and the phenotypes, the disease states, are known. And this method, called Mendelian randomization, can be used to combine genomic data, circulating proteome levels, and the phenotypes for new drug discovery. Truly exciting. After 9 months of exclusivity, the pharma partners have agreed, right, to make all their data public. And in terms of current timing, it's now April of 2022. By the end of 2023, the end of next year, you're going to see a remarkable explosion coming out of this particular project, combining genomics, combining proteomics, and combining a combination, a wide variety of disease states to truly understand, right, opportunities for precise targeted treatment. Why this matters? This matters because we have a faster and broader discovery of new drug targets that are likely to succeed. This matters to significantly reduce the cost and time to market in drug development, and it's going to benefit humanity and the future of healthcare as patients get access to efficient drugs faster. I want to thank you very much for your time and attention. Be happy to entertain any questions or comments, and I appreciate it.